ways in which a neuron finds its location. The first one's called pathway selection. This is fairly broad, meaning it's not very specific. It's say, hey, I'm here in Utah and I want to get to San Diego, I'll take I-15. That's it. You know, it's a very broad set of directions. Just go that way. Okay? So that's what pathway selection is you migrate along a route, but it's not knowing exactly, you know, exactly what it's going to do once it gets there. That's the first stage. The second stage is, okay, now I'm in the city of San Diego. I say San Diego because that's where I'm from. Now I want to find what street that I live on. That's where we get into target selection. So now you're in the city, or in the case of the neuron, you're in the general location of the cells. Now it needs to start finding what set of cells it needs to associate with. Is it going to associate with you know, muscle cells? Is it going to associate with cartilage? Is it going to associate with skin? Is it going to associate? What is it going to associate with? The last one, and this is the one where you really get into precise chemo factors such as proteins and receptors, is the address selection. Now you're on the street where you know you want to get to. Now you need to know exactly what cell that neuron is going to connect with. So this is where the refinement process comes into play. In fact, in most cases, you'll get several neurons reaching a certain target cell, but only one will end up staying with that and the other ones atrophy or die off. If you remember, when we talked about apoptosis, you actually generate way more neurons than you actually use because a lot of these will die off in the process of the neurons selecting their particular target. So that's the three modes. The pathway and target selection, these are typically done through a series of repulsions where the neurons are pushed this way or that way. The address selection has a very precise, exact arrangement of which neuron is binding to which cells. And this is, in fact, where we get into things such as Huntington's disease and Parkinson's disease, is because many of these neurons, in order to stay with their targets, need this constant reinforcement of the signal. And in the cases of some of these diseases, these proteins stop being expressed, the neurons atrophy, and they die off. And that's happening even in an adult, not just in the early stages of neuron development. This is illustrating the same thing from the PowerPoint, but I want to point out something here. So here we have some motor neurons that are expressing these genes. When they project out to the developing limb bud, which will be the next lecture we talk about, the main thing that causes this to come here is the fact that as it projects out in this general direction, it's not actually being attracted to the limb. It's just pushing itself out, and it's repelled by these epherin proteins. And so, in fact, this will go to this area. Again, this is where the pathway selection comes into play, is that this is choosing a path in which the neuron's going to grow. Well, this path is pushing it out this way, and if it tries to go down here, it gets pushed up into here in the dorsal region of the developing limb. The same thing is true for this neuron, except because it's exposed to a different set of genes, it's going to have a different set of receptors that are going to make it that it's repelled by a protein called semaphorin, and that will push it into the ventral region of the developing limb. So you can see it's not very specific in the beginning. It just says they grow out and says, well, okay, I'm going here because I'm repelled by the signals by this one, and this one's going to go here because it's repelled by the signals there. However, in some cases, as you can see in these other set of neurons right here, they actually have to be go to their area by a chemoattractant. And in this situation, you can see it makes a hard left and goes right up here to this mesoderm area. It can't do that except through attraction. And so these are the two main mechanisms that drive neurons to where they're supposed to go. It's either repulsion, to push them away from a particular area, or direct attraction. So what is it that's actually doing the repelling or the attraction? It comes down to the growth cone. There's a growth cone at the end of the axon that has these microspikes, which are part of the actin, or the cytoskeleton, of the neuron. And there are receptors on the surface of the cells that when they come in contact with certain ligands, it either causes these microspikes to be stabilized, and therefore the growth cone will continue towards that region, or it causes them to atrophy, and therefore it will be repelled and go in the other direction. So you can see how these microspikes just branch out, and depending upon if it's an attraction in this area, these will collapse, and then the axon will move that way. If it's a repulsion, like over here, it'll collapse, and these will go that way. So that's really how axons start moving to their respective sites, is either the microspikes are stabilized by that attraction, chemoattractant, or they break down because of the repulsion of the interaction between the proteins and the receptors. So here you're seeing the pioneer nerve fibers. You have this leading process that typically can have a chemoattractant, and this allows the subsequent migrating neurons to just follow that path. They're like, hey, well, here's the trail. I'm just going to follow that trail. They don't follow the same repulsion attraction that the pioneering neurons do. So that's where this comes into play, is that the pioneering neurons play a key role in making sure that the other neurons can get to where they're supposed to be. Okay? And that's where these come into play, the attraction that causes it to make a hard left to this muscle tissue here, or these. Now, what is it that ultimately makes it so that these will go to this tissue as well? Well, there's two main genes. Along the anterior-posterior axis, it's the Hux genes that determine where they're going to go. Along the dorsal-ventral axis, it's a group of genes which we call the limb genes. Here's one of them where you see limb 3. So make sure you know that. This is one of those factors in specification of these motor neurons, is that along the anterior-posterior axis, it's the Hux genes that help determine which target they're going to receive. And along the dorsal-ventral axis, it's the limb genes that are turned on. You can see here, limb 1 is in these, limb 3 is in these, and these are the genes that ultimately determine whether or not the receptors on the growth cones are going to be attracted to things like, or are repelled by things like semaphorin, or repelled by things such as ephrin. So here are some of the major proteins that we're going to talk about that are involved in chemoattractant and chemorepulsion of the growth cones of the axons of neurons. I've already talked a little bit about ephrins and semaphorins. We're going to talk about netrins today. We're going to talk about slit and robo. And then, of course, cell adhesion molecules, or CAMs. So let's look at the dorsal root ganglion. The dorsal root ganglion are these neurons that originate from the trunk neural crest cell that will project their axons into the spinal column and then outward to other respective tissues. These are the sensory neurons. These are the ones that are receiving information from your internal organs as well as from your skin and your muscles, telling you what's going on. Okay, so the dorsal root ganglion are the neurons that receive the sensory information. As such, they form in these clusters near the dorsal end of the neural tube. They'll project their axons out to the skin and the muscle and these tissues, but they'll also project axons into the neural tube because they've got to connect the two. They've got to connect the sensory information with their receptors in your skin to the brain. Okay? So how do they do that? Well, we talked about semaphorins. We talked about ephrins and things of that sort. One of the things that you should know is that there are respective gradients of these that will determine how far into a particular region these axons will go. So in some cases, it's not just about attracting them to it, but it's attracting them to a certain point. And that all comes down to the concentration of these proteins. So typically what happens is when these neurons are projecting in, some of these neurons have so many receptors that they're so sensitive to proteins that at low levels, once they reach a certain threshold, then they're repelled and they don't go any further in. Others have fewer receptors, so they'll go a little bit further in. And others have fewer receptors even still, so it takes a huge amount of this gradient to repel them. So essentially, these will move in and start synapsing at various regions depending upon how many receptors are actually in the growth cone that determine at what point are they going to stop moving inwards, and they say, well, okay, you found your spot. Okay, so in this, as well as you'll see in others, this is the case. Now, some dorsal root ganglion synapse with what we call interneurons. Interneurons are neurons that will then go straight up your spinal column into your brain. These are the ones that require cerebral processing to understand what's going on, the more complex reflexes that you have. For example, if you burn your hand or something like that, there's a complex withdrawal reflex. However, there are simple reflexes like when the doctor hits your knee. You know, when you go in and you're saying you're having headaches or other problems, why do you think that they hit your knee and other parts? Are they just doing it for fun? It's kind of it's a way of testing whether or not your reflexes work. They're testing your nervous system, essentially, and the ability for your nervous system to respond appropriately. So some of these neurons can actually synapse directly with the motor neurons. That's how your knee-jerk reflex works, is that you'll hit a stretch receptor right here in this muscle, and that'll synapse directly with the motor neuron, which will cause us to contract, and that's what causes your leg to go up. Others, like burning your hand, stepping on a nail, require a little bit more complex series of reactions, because when you step on a nail, you're not just pulling your foot away, you're also balancing on your other foot. It's a very complex series of reactions that have to occur, which require integration into your brain. So that's why a lot of these will synapse with these interneurons up here and not directly with your motor neurons, so that you can have some sensory processing here, and then that can influence other tissues that aren't directly involved with that reflex. So let's talk about some of these interneurons. One group of them are called commissural neurons. The commissural neurons are very interesting, and these are a good example of the intricate interplay between attraction and repulsion of growth cones. Let me explain what happens. Commissural neurons project downwards towards the ventral neural tubes, then they cross it, and then they project up to your brain. So in fact, these neurons will synapse with this side of the body, they'll cross the spinal column and go up to the left side of your brain. And things from your right side of your brain synapse from the left side and go up there. So you have this crosstalk, even in your spinal column, from the left and the right hemispheres as it goes across the spinal column. So the question becomes, how does the neuron know first to go towards here, and then to cross here, and then to go up to the brain? Well, they found a couple of genes that are responsible for this. Sonic hedgehog being one of them. We already know that that's being expressed in a gradient-like fashion along the ventral neural tube. But there's also another protein called netrin. And these netrin proteins are a chemoattractant to these commissural neurons. So these neurons already develop the receptors that allow them to be attracted to this particular protein. Once they cross this midline, however, they have to then receive a repulsion to be pushed away and then up to the brain. And that's a very complex process. Well, that typically requires that things change genetically in the neuron so that it's no longer attracted to here. Because otherwise, this would come all the way to here and just stay right there. It wouldn't cross the midline, and it wouldn't go upwards. So let's look at an example of how this works. There's a couple of proteins and receptors that are in this that I want you to explain to me. So let me go through how this works. Along the midline of the neuron, you have a group of proteins which we call SLIT. And these proteins repel these axons. And this is why they don't cross early, is because as they're attracted to netrin, they won't go near this midline. They're actually repelled by it, so that as they're attracted, they'll just move right down along the periphery here. But there comes a point where they need to not be repelled by this midline anymore. So what happens is they are, when they get close enough, certain genes get turned on, which will actually prevent this chemorepellent. So here's an example. SLIT will bind a robo, and this keeps a receptor called DCC downregulated. It prevents the transcription of this gene. When this gets close enough, however, robo1 gets blocked. Now it's not repelled by SLIT anymore. And since robo1 gets blocked, guess what? Now DCC receptor gets turned on. Netrin attracts it. It moves across this region. And then guess what? Robo1 turns back on again, and it repels it back away from the midline. So there's actually receptors that are going down and others that come up, and then it goes back to the way it was, so that you get this very complex series of attraction and repulsion to allow these neurons to be able to cross the midline. So once it crosses this midline, Robo1, it gets turned back on again, which then blocks DCC, and then it gets repelled. So once it gets here, then it goes back to the way it was, and that pushes it away from that midline, and then other attractants and repellents are going to push it up to the brain, and it's going to find its location up in the brain as it's developing in that region. Let me go over this again. Initially, you have the receptor, Robo1, in these commissural neurons. Slit is all along this neural tube middle region right here. This prevents the neurons from actually crossing over the lumen at this particular time. It makes it so that they keep going down all the way to the foreplate. Okay? So as long as Slit and Robo are attaching to one another, this will not be able to cross the midline. However, as it gets closer, there are other genes we're not talking about here, but as it gets closer, these other genes will cause Robo3, which is a different protein, to prevent Robo1 from being made. That then takes away that repulsion. Well, now that this is gone, this protein can now be expressed. Netrin causes this to be attracted, moves across, and then another series of proteins reverts it back to the way it was. Robo1's expressed, and then it repels and pushes it past that midline again. So it's just an illustration of the complexity of the neuron finding its area. At this point, it's still not quite sure where its target's at. This is pathway selection. Okay? This is where it's just saying, oh, I'm not supposed to go down here. I'm just supposed to go this way. Oh, now I'm supposed to go across here. It hasn't found even its target yet, but it's finding its pathway. So this is the very nondescript or very general way in which these pioneering neurons are going to be moved to their location. Let me show you another example, limb development and such. So here we have a neuron that is repelled by semaphorin. As soon as it reaches this point, it can't cross that midline. So guess what? It starts moving where it can. Well, it gets to a point where they've put some beads that are secreting this gene, and that's why they showed that this gene X is an important factor. When this gene synapses with, or these proteins synapse with some receptors, that then removes the repulsion of semaphorin and allows this to cross across and then go to its respective target. So again, it's the same concept as the commissural neurons. It's repelled. Some other protein is then going to cause a change in that repulsion receptor combination, and then it allows it to cross where it's supposed to go, and then it will continue on down its path to its particular area. Same thing true. Again, there's just tons of examples of this. With the retina, as it comes from the optic disc, it then has to decide whether it's going to go around this side or this side. These have to do with both attraction and repulsion factors. Eventually, it'll reach this point. Again, when we get into the area, once they're at that path, they're in the city, they know that they're at the right city, then we get into what we call target selection. Remember, target selection is what street do you live on. And this is where you get this process. And I explained this before with the neural tube, but here's another example of how the, you get it from the eye to the brain. Is you get these gradients of things like ephrin. So here's an ephrin ligand. It's a very high concentration in the posterior end, very low concentration in the anterior end of the optic tectum. Well, as the neurons are projecting in, each one originates from a different region. This one originates from the temporal region that will go with the temporal lobe. This is from the other region. Notice that the ephrin receptors, this is an illustration of how many there are. If there are fewer receptors, it has to go further into the gradient before it finally receives that repulsion. And that's what allows this to go deep into the posterior end. The more receptors it has, the less of the ligand it needs to activate that repulsion. So that's why in these, it's just about amount of receptor, really, is whether or not it goes a little bit into it, a little further into it, or really far into it in terms of how much ligand-receptor combination is needed to initiate that repulsion of the axon so that it doesn't go any further into that particular area. Here's just an example showing they did some different things where they had repulsion, where they had high levels of ephrine versus low levels of ephrine, just showing you how these migrate along these different paths. In fact, that's how we get neurons to go where we want them to go, is typically through repulsion factors of different gradients. So this is just showing the same example of the differential concentrations of ephrine. Now let me talk a little bit about, where is it, address selection, because this gets into Parkinson's disease and Huntington's and Alzheimer's and all these kind of stuff. Let's talk about Huntington's disease. Once the neuron reaches its exact target cell, these cells typically secrete factors or proteins that allow the neuron to survive. Okay? Some of the neurons in your brain release certain what we call neurotrophic factors. There's brain-derived, there's glial, there's, what else is there? Make sure I have these highlighted. There's so many of them. Conserved dopamine, neurotrophic factor. Let's talk about brain-derived. This is where Huntington's disease comes into play. In order for some of the neurons that influence Huntington's disease to be maintained, they need this brain-derived neurotrophic factor to keep the neurons alive. And this happens throughout your whole life. Essentially, to keep that connection, there must be some reciprocal induction of the target cells. So this same address selection that occurs in the early stages of development is maintained through the life of the cells. Well, what happens in Huntington's disease is there is a protein that we call Huntingtin, not Huntington after the person, but Huntingtin, that regulates the amount of brain-derived neurotrophic factor in certain neurons in your brain. Well, guess what happens? As the person gets older and you have less and less of this sustaining, neurons start to atrophy, and that's where you get the motor and the memory and the other problems that are associated with Huntington's disease. We do not have a cure for this, but we do have drugs that can try to maintain this brain-derived neurotrophic factor stable for as long as we can. So that's where a lot of these drugs for Huntington's disease come in today, is to keep those neurons surviving, even though Huntingtin is downregulated and can't keep that alive. Let's talk about Parkinson's. Parkinson's comes about primarily from a downregulation of a dopamine neurotrophic factor, and that's what causes a lot of those neurons to atrophy. Alzheimer's and Down syndrome actually have a similar protein in common. It's called the AP protein, not like a phone app, but it's APP. The AP protein, here's what happens, is when a neuron reaches some of these target cells that influence Alzheimer's, it's not the only protein that influences Alzheimer's, but there's a protein that's released that is absorbed by the neurons that then go all the way back up to the nucleus and then tell the cell certain genes to turn on. Well, there's a deficiency, there's a protein called the AP protein that blocks the ability for this neurotrophic factor to be pulled up into the neuron, and then again the neuron essentially dies and atrophies, and this is what causes a lot of the degeneration of neurons both in Alzheimer's and in Down syndrome. This protein is actually found on chromosome 21, which is why when you get trisomy 21, this causes Down syndrome, because it influences the neurons. You have more of this AP protein, you get less of the neurons being able to integrate this neurotrophic factor into their cells, and the cells won't express the right genes, and the neurons atrophy and die. So this is the reason why we need to understand how neurons not only find their location, which gets into neuron regeneration for spinal cord injury, but to help with diseases like Huntington's and Alzheimer's and Parkinson's that we don't have any cure for, because once the neurons atrophy and die, we can't get them back. We can't help them find their location again, and so the only thing we can do is delay the onset of that by causing these neurons to be able to have this neurotrophic factor, whether it's brain-derived or dopamine or whatever the case may be. So that's where a lot of research is going into today for these, is to not only understand the developmental process of them, which continue on throughout your whole adult life, But how can we, once you're an adult, go through the same process to maybe get those neurons back to their connections? As you can see, it's a very difficult process, even in the earliest stages of development, for these neurons to find their muscle group, then to find which group within that muscle that they're going to target. Your muscles actually look, when you have these muscle fibers that synapse them, it doesn't synapse it in like one part of the muscle. It actually spreads out and has these clusters so that the whole muscle can contract evenly instead of more to one side than the other. And it's a very difficult process to even get that type of randomization and the selection across the whole fiber rather than having these clusters just in different parts of the whole organ itself.